Good afternoon to everyone. So I am Denis Rivière. I'm working in NeuroSpin in France. And we are developing for a long time now a software suite which is called BrainVisa, which is freely available on the website brainvisa.info. And I will demonstrate today the use of the Sulcal morphometry toolbox, how to use morphometry of the cortical sulci for multiple subjects using this toolbox. BrainVisa contains a number of different toolboxes. Morphologist is the main toolbox of BrainVisa, which is developed by our team, which is dedicated to the study of cortical folding and cortical sulci analysis. BrainVisa is distributed now as a virtual container or a virtual machine. It's running on natively on Linux systems, but it can run on other systems using a virtual machine. We have put a little demo dataset at this address, brainvisa.info/download/data. And there are actually two different datasets here. One is a complete, sorry, I won't use the mouse after all. Complete dataset containing the BrainVisa distribution on virtual container and a few T1 MRI images, which are actually extracted from the Human Connectome Project public dataset. It's 8.5 gigabytes, so it's a bit large. If you want a lighter dataset, you have just the raw data here in the second dataset. So in this archive, you have the dataset plus the slides which I am presenting here and a few scripts which I will be showing today. So if I get to more hands-on, so BrainVisa is run through a virtual container, so you can start the container once it's installed. And inside this container, you can run either a program like BrainVisa, Morphologist, or a bash shell, which I am going to do here. So here I am in the container, and I show things here. So the goal of the toolbox is, as I said, to study cortical sulci morphology, and it allows to start from raw T1 MRI anatomical images to segment the tissues and extract the cortical folds. Then there is a sulci identification step, which allows to assign a name for each sulcus, which are visible here with colors. Each color corresponds to a specific sulcus. And then you can conduct inter-subject studies by comparing morphometric features measured on these folds. There are different ways to operate the toolbox, actually. So the toolbox contains mainly one main pipeline, which is called Morphologist. We have a graphical user interface, which is called Morphologist UI, which is a simplified application, which is easy to use when things go right and when there is no problem, when the image quality is okay, etc. And which doesn't have any parameters, tuning, etc. It's straight. There's another graphical way, which is a more classical way, using BrainVisa. BrainVisa is a kind of container for many processing tools, and there are different toolboxes. So Morphologist is just one of them, which show how to use it also. And there are also ways to process data using non-graphical tools, using either command lines or programming using Python IP. BrainVisa is written in Python. So I will show you first how to use the simplified user interface. Everything is in the BrainVisa container. It's run using the program Morphologist, compared to the classical version, which is maybe more known to users. It's simpler to use. It doesn't use any database indexing, contrarily to the classical version. There's no parameters tweaking, as I said, so no possibility of correction if things go wrong. But it's still compatible with the classical BrainVisa version. The data layout is the same, files are written at the same place. And so it's possible to use BrainVisa after Morphologist interface. It's able to process multiple subjects, but maybe not hundreds or thousands of subjects, because it was designed quite a long time ago, and the size of the data sets we had to process was not as large as today. So I will show you in practice. So I will run the program Morphologist. So it starts a graphical interface, which looks like this. So it shows a number of empty windows for now, which will be filled when there are data to display. To process data, I must everyone sees, is it big enough? So I have to create a new study, defining mainly data directory, import images. So I will create a new study. I can give it a name. I will process some HCP data, so let's call it HCP. I have to choose a study directory. I can create a new one. Let's call it morpho data. And then I can choose the formats of the files which will be written. Let's say compressed NIFTY and GIFTY for meshes. Then I can import raw data, which will get from files. In the little data sets that I have shown, there's a raw data directory where I can take images. I have prepared 10 different images, but for the demo I will use only 4, because I will run on my little laptop, and it will take a bit of time to process. So we'll take only 4. So I see the images which will be imported. I can, if I want, change the name of the subject, or change the group name. By default, it's called undef. I can change the group. Let's say HCP. Then I can apply. It will import the data. I can see the details. While importing, it will check that data are valid, check the voxel type, the intensity ranges, and apply some conversion if needed. And the data will be written in the study directory. When it's done, I can close. And you see then that the raw image appears in the first display window. So this is an interactive display. We can change the orientation. You also have a more complete visualization when I click on the upper right corner here. So you have the 3 classical slices plus the slice in space, in 3D space. And every display will have such a spatialized view. The other display will be filled whenever the data are processed. Here on the right, you have the list of subjects and can switch to another subject, and the data will be loaded and displayed for that subject. When it's done, I can run the processing. As I said, this application runs a single Morphologist pipeline, so there's no option. I just have to run. So when it started, you see this little rolling button which moves and shows that things are running. And in the status bar here, you see which step is currently running. The first one is a normalization step, which is based actually on the SPM software. And it will process further. It takes a bit of time to process on my laptop especially. So while it's running, I will explain other things. If I get back to the slides are also in the dataset which is distributed. There's a tool to have a better monitoring of what is running, which is an external program, which is still part of the BrainVisa software, which is our pipelining on distributed processing tool, which is called Soma Workflow. And there's a program called Soma Workflow GUI, which can be run. If I start it, I see here there's one workflow running, showing one pipeline for each subject. In each pipeline, there are different steps. If I can expand all, so you see all the steps which have to be processed. Some steps have already run, others are running, and the others are waiting for their dependencies to be met. There are some graphical views of the number of jobs which have been run. So this tool is able to process either locally on a local computer or to connect to a computing resource. In this mode, you have to configure the computing resource. I'll go back to the Morphologist. So there's not yet any other data to display, so I'll come back later. In the meantime, I can show you how to run the other graphical way to run Morphologist using the classical BrainVisa application. So BrainVisa is also a program that you can run in the BrainVisa container. It takes a little while to start because it's loading lots of toolboxes. So it looks like this. So this is the main interface of BrainVisa. So you have a list of toolboxes on the left panel. Morphologist is one of them. And when you click on a toolbox, you have a list of processes which can be run. Other processes are located in subdirectories. So there are a lot of available processes. Before running Morphologist in BrainVisa, as in the graphical version, you have to define some data directories, which are called databases in BrainVisa. So you have to go to the preferences in the databases tab, and you can add a new database, which is basically a new directory. You can select either a new directory or an existing database directory. And actually, I can use the directory which I have already created for the simplified application. There are other settings that can be chosen, but here I will go with the default. And once it's defined, I can validate. And then I have the opportunity to update the database. This means that all files in the directory will be scanned and indexed in a little database, which allows to make queries. I will do it because the simplified application doesn't use it. It's a very small database, so it's fast here. And then I can go to the Morphologist toolbox. And the main pipeline is Morphologist 2021. But before I can run it, I have to import the data. In the import directory, there's the import T1 MRI process, which I can open by double-clicking it. All processes in BrainVisa have this kind of interface. They have a list of parameters and an empty space at the bottom, which is reserved for when things are running, information is displayed here. On parameters, file parameters, you have different buttons. One is for getting files. You see the cursor here. So this directory icon is to get files directly on the file system. And these green and red disk buttons are reserved for accessing via the database. To import data, I will get data out which are not already in the database, so I will use the raw file system button. And we get the data in the raw data directory. I can select several, sorry. Here, this process is dedicated to one image. So I will select one image. And then I can select where the output will be written in the database. So I will use the database button, which is red for output files, which will be written, and red for input files, which will be read. Here it's an output. So I can select the database. I have only one, so it's easy. I can, if I want to select the same data as the simplified application, I will use the same center parameters. These are actually some attributes used for indexing. The file format, let's use compressed nifty. And I can select the output. Now if I run this process, the file will be read, converted, and written in the output database. I won't actually run it because it has already been done using the graphical application. As you see, data have been processed in the meantime. I will come back to it later. There's a possibility to iterate any process in BrainVisa using here the right menu button. And I have to click on iterate. And every parameter is transformed into a list. So I can select here, it's the same parameters, but I can select several ones. So I can go to the raw data directory and select maybe the 4 images. Okay, I can also select the outputs the same way, using the same database. Nifty compressed format, and I can select a list of output images. When I validate this, I get a pipeline, which each step is one instance of the import process. So I won't run it either because things are already running. So I will just get back to the graphical application. As you see, it has processed some new data. This is a bias-corrected image, so it has been corrected for the non-uniformities of the signal. And then there's a histogram analysis to detect the intensities of the white matter and gray matter intensity peaks in the T1 MRI image. Then there's the brain segmentation. On each view, you have a more detailed view, which is also interactive. Here I have the gray and white matter segmentation, also with the spatialized view. And things are still going on. I can switch to another subject while it's running, so it will read the data for this subject and display what have been already produced. So I will let it continue to process, and I will get back to the BrainVisa interface. So now, let's say I have imported the data. I can run the Morphologist pipeline, which is the first one here at the top. Maybe, is it large enough for everyone to read? So if I double-click the process, I will have a view. It gets a bit of time to open, especially because my computer is already running. Here it is. So the process looks a little bit like the import process, except that there's a left panel here showing the different steps of the pipeline. The pipeline is actually a list or a tree of processes, which will run with dependencies between them. Each process has its own parameters, which are mainly summarized in the main pipeline. So the main input is a raw T1 MRI, which should have been imported in the database. So I will get it using the green database button. Here in the morphodata database, I see different images. I can select one. And then the different outputs will be filled using parameters completion system, which is included in BrainVisa. So all these steps will be run if I click on the Run button. There are two ways to run BrainVisa process, either using the Run button, which will run everything in a single processing sequential way, or using the Run in Parallel button, which will make use of the Soma workflow tool, which will use distributed processing either locally or connected to a computing resource. I won't run it this way because, again, it's already running. You can iterate the pipeline in the same way as you can iterate the importation either by clicking here the iterate button from an already open process or from the process list here. And then you select a list of T1 MRI images. BrainVisa has some bottlenecks, it's convenient to run a few dozen subjects, but when you get to very large databases, it becomes very slow. So we generally use it to run maybe 100 subjects, not more. There's another interesting process here, which is a Morphologist QC table, which displays the data which are actually available in a given database. If I run it, I get a table of the data which are present in the database, which has been produced, and the data which should be produced and which are missing. If I double-click on the data here, it triggers a viewer process, which is an interactive view of the data. Most views are done using the Anatomist software, which is part of the BrainVisa distribution, which is a 3D viewer dedicated to neuroimaging data, which is able to display all kinds of neuroimaging data. Here, there are two images superimposed, but it's able to display meshes, fiber tracks, and lots of other data. There's a slide at the end of this presentation displaying a few scenes that can be built using Anatomist. So it's a very general tool, actually. It can be used standalone. You can run the Anatomist application, and it also has a Python API, so you can use it in scripts or programs. And Anatomist is also the viewer which is used here in this views in the Morphologist tool. So here you see that meshes have been produced. The gray-white matter interface and the pair mesh together with a slice. If you get to the specialized view here, you can see the intersection of the meshes with the image, so you can check that the segmentation is correct or not. It's an interactive view. You can change the slice and click also. So this viewer principle in BrainVisa is quite of in general also. So you can open several viewers at the same time, of course. This is a view of the non-uniformity corrections, bias correction. It displays, so it's a spatialized viewer, also based on Anatomist, and it displays the raw image and the corrected image using a color table, which allows to see if the intensity of the gray and white matters have been homogeneized along the image. You can see different orientations. Let's get back to where we were. So this was BrainVisa processing. There are also command line options to run Morphologist. Actually, Every process in BrainVisa can be run using a command line tool. It's a program called Axon Run Process. I will close a few windows. So I can use .h parameter to have the help of the command. And this is a general command which is able to run any process in BrainVisa. So Morphologist is just one of them. There are many others. And there are a number of parameters in order to, for instance, enable or disable databases capabilities. Using databasing in BrainVisa slows things down because it makes many requests, especially to perform the parameters completion, to generate the output file names for the process. But if you don't enable it, there will be no completion, so you have to specify every parameter of the process, which is, for Morphologist, you just don't do it. So you will use NML database. There's an option to use the some overflow distribution system or not. By default, it's not used. And another important parameter is the iteration feature that will tell the program that you want to not to run on one image, but to iterate on a series of images. So you can specify which parameters will be iterated. Here, for the Morphologist tool, pipeline, it will be the main T1 MRI parameter. It's the same parameter as you see in the graphical interface. Maybe I've closed it. So T1 MRI. So typically you can run Axon Run Process with option Enable DB. I will iterate using some overflow. I will iterate the parameter T1 MRI for the process Morphologist. And then I have to give the values of the iterated parameters. So it's not one file here, it's a list. And I have to specify the list in a Python syntax, which is quite similar as JSON, using these braces. I won't run it actually because once again it's already running. It's just to show how it works. Let's say image1.nii.gz. And if I want to run on a second image, so this is how the general Axon Run Process command works. So it can be called from a terminal or in a script, etc. So I am not running it actually. I'm just checking that things are going well here. So it's processing the cortical sulci. It's in the process of extracting the cortical folds. My little computer is quite busy, so it takes a bit of time to update. Here it is. Here the folds have already been extracted, as you see here. So the folds are virtual surfaces going inside the folds of the cortex. They are actually in the CSF fluid, so they are not real objects, but they are used to represent the folds, and these are actually the objects which are used for morphometry. So the brain folding is highly variable, and so there are lots of differences between subjects. And so then the next step will be to identify and give a name to each of these folds. In the spatialized view here, I can see this same 3D view. And in the 2D views, I see the intersection of the folds with the 2D slices. Of course, I can change the slices. It's a bit slow here. Etc. So I will let it run. So let's go back to the presentation. So as I said, the BrainVisa pipelining infrastructure has some limitations. When you run some very large databases, even the command line interface, we generally process batches of about 500 subjects at one time. There's another way, I just mentioned it, but I won't actually show it. We have designed a new pipelining infrastructure to try to get rid of the bottlenecks of the older one. But it hasn't been integrated in the graphical interfaces yet. This new infrastructure is called Capsule. It's a Python library, and it also has a command line interface. So it's very similar to Axon run process. You're just calling using Python command and .m capsule. And then it's almost the same parameter. I can get help. And the same pipeline is available in the capsule infrastructure. Sorry. It's almost the same parameters, except for the iteration. It's a capital I instead of lowercase I, because the lowercase I is used for specifying the input directory for data. As it's not using a database, you have to specify input and output directories. Generally, it's the same, but you have to specify them. So this pipelining infrastructure is also running through the SOMO workflow distribution system. It has no database indexing. And so the initialization time, which is the main bottleneck in BrainVisa. So in Capsule, it's the initialization time is linear with the number of subjects you are running. So you can process more subjects, but still it's not perfect. After a number of subjects, it also begins to be slow. So it's still processing. There is also a Python programming interface to use BrainVisa, which is displayed here. It's a bit smaller. It's written larger here. So you have to import a few libraries, and there's an initialization to be done, because at the beginning, more than 20 years ago, BrainVisa was designed as an application, not as a library. And so there are some internal global state variables to be initialized. So you have to call this initialize processes function before things are actually set up. And then you can import other functions. And to run a process, things are triggered through this context object, which basically links between the processing and the outside world, like where the outputs of the processes will be written, either in the terminal outputs or in a graphical window, etc. And basically, it has a function run process. You tell it the process to be run, the parameters, and it's quite similar to the command line interface. Or you can also instantiate a given process using the getProcess instance function. Here you can instantiate the Morphologist pipeline, but you can get any process in BrainVisa. And then this process object has the parameters. You can also tune it by selecting or unselecting the processing steps in the pipeline. You can change things, and then you can run it using the run process function of the context. So it's still processing. Then, when the processing is performed, the last step of the Morphologist pipeline is the feature extraction on identified Sulcal. If you see the list of steps in the BrainVisa pipeline, it's the last step, which takes some measurements on the cortical folds individually, sulcus by sulcus. It can measure the length of each sulcus, the average depth or maximum depth, the average cortical thickness around the sulcus, the sulcus opening, which is the thickness of the CSF fluid inside the fold, which is a marker of aging or atrophy. And all these figures can be used to perform morphometry studies. So here it hasn't finished the processing, but when it's done, you can use the menu here, morphometry, and then you can export all the measurements of the features in a CSV file. I can't do it yet because it's still running. And also, anyway, I've processed only 4 subjects, and as you know, the Sulcal variability is very wide, so you can perform this kind of studies on a quite large number of subjects. These images are extracted from the Human Connectome Project data set, and we have already processed all the subjects. And in the data set that we have provided, we have put a CSV with all the subjects of the HCP cohort, which is hcp-morpho.csv. So to get these measurements, as I said, you can use it. You can do it using the simplified Morphologist graphical interface. as I have shown, or in BrainVisa, this can be done using inter-subject morphometry process. There are several ones, actually. I will show two, depending on the shape of the files you want to get at the end. It's not very different, actually. So one is still in the Morphologist toolbox, of course. In tools, there is a process called create an inter-subject measure file. And this is a process which is actually run in the simplified interface. So you select, using the database, the individual measurement files. Here there are none available yet. And you can select an output CSV file, and it will write inside it. An alternative to get the inter-subject measurement is in the morphometry subdirectory in Sulcal. You have a Sulcal graph morphometry inter-subject. This one is very similar to the other, except that it will allow to select only one measurement, for instance, Sulcal opening or the average depth, either in a native space. No, it's all in a. It's in normalized space, Telerak. And you select not a file but a directory, because it will write one file per sulcus. So it's smaller files if you want to select later, but it's the same numbers in it. Actually, there was another morphometry features extraction process, which is older but a bit more difficult to use, which also is in the morphometry subdirectory. It's called morphometry statistics. I just show you that it exists, but it's more tricky to use because you need a model which will tell how to extract the features. And it's able to extract a lot more features, like the average position of each circle, its orientation, the number of connected components, and also relations between pairs of sulci. So if you need it, it's available here, but I won't show how it runs today. Here you see that the right hemisphere of both hemispheres, actually for this subject, have been processed. And so now you get the colors of the sulci. Oh, it's slow. Computer is still working. Normally it's not so slow. Well, here it's okay. So each color corresponds to a sulcus name. Here it's a superior frontal sulcus. Here in red it's the central sulcus, etc. In the detail view here, you also have both 3D and 2D views of the sulci, with colors corresponding to the sulci names. I'm sorry, it's a bit slow. There are tooltips displaying the name of the Saclay also. You can also extract the intersubject morphometric features using command lines, using the same commands as for running the Morphologist pipeline. Just the name of the process and the parameters which are different. If you want to get the name, the identifier of a process, you can get it in the graphical interface. For instance, this one. If I click on a process, I didn't tell it, but you have the documentation on the right panel here. And in the technical information, you have the file name of the source code of the process. You can have the source code if you need. But especially, the file name is actually the identifier, the short file name. So here it's morphometry. If I get this one, which create intersubject measure file, it's concatenate files, etc. So this is the name you are using when you run the command line, which is shown here. Once this is done, you have a CSV file, and actually it's the end of the real BrainVisa processing. Our toolbox stops here, and then you are free to use the tools you like to process the statistics to, for instance, compare different populations, etc. So this can be done using any statistics tool you like. I can show you a little demo of how to manipulate this kind of figure using Python language and standard packages like pandas, numpy, scipy, and matplotlib for display. On the dataset archive, you will find a little script which corresponds to this little program actually, and a notebook which I will show you, which is here, which is just to demonstrate what kind of thing you can do using Sulcal morphometry. It's very simple. You can do very more advanced things, and I'm sure most of you are more expert in statistics than I am. So I will run this notebook. So the first cell is just to import the different modules in Python. Then I will read the morphometry file for the full HCP dataset, which includes 1,100 and a few subjects. I don't remember the exact number. And for this little demo, we will focus on the superior temporal sulcus, just on one sulcus, to try to get measurements on this sulcus on both sides, both hemispheres. So I'm using the pandas libraries to select things. So the table looks this way. So you have one line per subject. Sorry, two lines because there are the sulcus, the left and right hemispheres, and then there are different columns for the different measurements. The measurements are available both in a telluride space, in normalized space, which are corrected from global size effects, and also in native space. We will also load a file which is provided with the HCP dataset, which is the participants file, which provides some information about the subjects, like age range, gender, etc. We'll not use much of it, just gender just for the demo. We merge both tables, one from the Sulcal morphometry and one for the participants. So I'm just using a pandas library function to merge the tables. And we will concentrate on the average depth of the sulcus, because we know that there's something to see on this depth on the temporal sulcus. So we'll use the column which is named mean depth in telluride space. Then I can plot using the matplotlib library the data. Oh. So I can do the same plot separated by hemisphere, because I have both hemispheres mixed. You don't see it, but here the left hemisphere is displayed in blue and the right hemisphere in red. Sorry, and you don't see it, but the blue dots are significantly lower than the red one, which means that the right hemisphere of the superior temporal sulcus is deeper than the left one, which is something which has already been described in the literature. We can also display histograms for that. So this one is okay. So it's the row plots which didn't display correctly. So the left hemisphere is in blue and the right hemisphere is in orange, and you see that there's a very clear difference. So it's possible to do stats using SciPy. Here it's just a t-test, and you have the p-value of the difference, which is very, very significant. If you get back to the potential gender effect, we can do the same stats and plots using the. Yes. For this specific sulcus, it's not the case for every sulcus. Yes, the superior temporal sulcus. So it's a histogram for the males in blue and females in orange. There's less difference, but there's still a difference, and we see that the left, the blue data are a bit lower than the orange one. This means that males have shallower temporal sulci than females. We can do. Sorry? It doesn't mean less gray matter, it means that the sulcus is less deep. In a normalized space, because they are measured here in a normalized space. Sorry, I've lost what was. Oh, sorry, it's for the left hemisphere and then for the right hemisphere. And, it's kind of similar. You have the same effect on both sides. And the statistics are still are actually significant. I skipped a little part. Oh, sorry. I was here. This is for both hemispheres mixed. And here, the statistics are very significant already. The p-value is in 3 exponent minus 10. If we separate the study by sulcus, by side, sorry, because the left and right hemispheres are kind of very different, we get these histograms. And the p-values are a bit, they are still very significant, but a bit less, maybe because there are less data for each hemisphere than for both. The last thing I want to display is that, as I said, this has been processed using the average depth of the temporal sulcus, superior temporal sulcus in a normalized space. If I use a native space, I can do the same. So this one, this time I select the mean depth native column of the data table. And I'm doing the same using both hemispheres. You see then that this time the blue data are a bit higher than the orange ones. This means that the effect is inverted. And the statistics, so it's not on the same side. The statistic is positive. It was negative in all the earlier tests. And it's still a bit significant, but far less. Which is probably due to the fact that male are taller than female, and so the brain is also bigger. And here it's not corrected for the global size effect. So when you perform a study, you have to be careful of this kind of effects, and you have to select if it's appropriate to use normalized or native space measurements. This is actually the end of my talk. We can process with questions if you have. Thank you for your attention.
Does it accept high resolution input, like less than 1 millimeter? And does it preserve high resolution input?
The processing was actually designed for image of about 1 millimeter resolution.
Okay.
Oh, sorry. Yes, you're right. So the processing has been designed for image of about 1 millimeter resolution, actually. It can process images of higher resolution. We sometimes process images of about 0.5 millimeter resolution. It works, but it's not optimal. It means that it takes far longer because the images are 8 times bigger. And also the Sulcal extraction is making quite often little bit more ramifications, which some of them are real, but not all of them, because some are segmentation artifacts. There are some parameters or thresholds inside the software that could be adjusted, should be adjusted, but we haven't done it yet.
Okay. I hope you might work on that. It'd be interesting. But, right, thanks.
The first one is you, of course, you use a label, you give names to the Saclay.
Yeah.
Where do you take the names? From where? I mean, which atlas do you use?
Good question.
And the other one is, which is, so you can do everything you describe. Oh, by the way, it is a beautiful tool. You can do what you want with the normal brain, but what about a stroke or a tumor or whatever?
So the first question is about how I define the Sulcal. We have a model of the Sulcal which has been trained by now it's a deep CNN network which performs the automatic identification, and it has been trained from the database of manually labeled Sulcal, which have been labeled by us, actually. We are working on this topic for more than 25 years, so we consider ourselves as kind of specialists of the sulcus anatomy, but we still make mistakes. We never have a certitude on the identification of Sulcal. So The automatic tools also makes mistakes sometimes. Not many, but it has about 90% agreement with our labelings. Now the nomenclature.
Sorry, you do not use the MNI standard brain, so the Sulcal that are.
No, it's a nomenclature which we have designed along years. Yes, and so this nomenclature has limitations, of course. We are not sure, especially in some regions which are more variable than others. And so this is why our research program now goes to trying to make some unsupervised learning to try to detect, I would say clusters to be to say it fast, but it's not clusters. It's patterns that can be found in subpopulations, but not necessarily in every subject. And so this is what we are working on the research side now, but this is not available in BrainVisa yet. And if you want to perform morphometry, Sulcal morphometry on other nomenclatures, so you're a bit limited because the model we provide has been learned on our nomenclature. It's possible to train it on another, but you have to provide a different data set of manually labeled Sulcal. And then what is possible is to group Sulcal. If you, for instance, the precentral sulcus in our nomenclature is separated in 5 different names. You can group them in a single one to have one measurement for the whole precentral sulcus, for instance. And for the second question, when you have a stroke, the segmentation tools actually don't perform so well. It's possible to provide a mask of the lesion in the Morphologist segmentation pipeline. So it will get this part apart and try to segment the rest. With more or less success, it doesn't always succeed. When it succeeds, sometimes it's okay outside the lesion, but it's a kind of crap inside the lesional region. So it's on a case by case basis.
You are getting there. So it's something that is in a program of your study. So I think you will develop a good tools for.
Well, the tools we provide are actually not really designed for pathological stroke subjects. So far. But and actually there are possibilities to use external tools for the tissue segmentation. You can use, for instance, FreeSurfer. And in the toolboxes in BrainVisa, there's a little toolbox to make a bridge between FreeSurfer and Morphologist. It's able to import the segmentations of FreeSurfer, write it in the Morphologist database, and then extract the Sulcal. So if FreeSurfer succeeds where Morphologist fails, if something happens, then you can still extract the Sulcal. But there's no guarantee that in the lesional region, everything will be okay.